Friends, welcome to Biology Made Easy. In the previous video, we have discussed the different aspects of linkage and the linkage groups, and today in this video, we'll discuss the remaining part of linkage and recombination. What are the different types of linkage, what is coupling repulsion, what is cis and trans arrangement, and how the chromosome maps are constructed. So let us start. First with the types of linkage. Types. Usually, the linkage is of two types. It may be complete or it may be incomplete. It may be complete linkage. So when we call the linkage as complete, when there is no recombination, there is no crossing over. That means crossing over is 0%, then linkage is 100%. So when it is possible, when the genes in the chromosome are very closely present, it is shows complete linkage. This complete linkage is very rare, but Morgan has reported it in male Drosophila. So the example is male Drosophila. Then the second one is incomplete linkage. Incomplete linkage. Now this, when we'll call it incomplete linkage, easily you can tell when the linkage is not 100%. That means when there is a recombination, when there is crossing over, then linkage decreases and recombination or crossing over increases. As already I have told you, that linkage ranges from 50% to 100%, while crossing over ranges from 0 to 50%. So as the linkage will decrease, the crossing over will increase. Usually, when we make a dihybrid cross, usually the different parental types and recombinants, they are obtained at 25% each. But in case of incomplete linkage, usually we will find that the parental types are more while the recombinants are less. So this is commonly found in case of sweet pea. In case of sweet pea, that is Lathyrus sativus, and also it is found in case of Drosophila. So this is that means the types of linkage. Then we discuss this another phenomenon, that is coupling and repulsion. According to Morgan, the coupling and repulsion are two sides of the crossing over, just as the two sides of a coin. So what is coupling and what is the repulsion? Now usually in coupling phase, let us discuss first the coupling phase. Coupling phase, what is happening and what is happening in case of repulsion phase. To know what is happening in coupling phase and repulsion phase, Morgan took some characters. So I am writing here the characters, the character, then dominant character and recessive character. So he took the flower color. Flower color, dominant is blue, so capital B, and the recessive is red, small b. And the other color is pollen shape, and the pollen shape long, it is the dominant, capital L, and round is small l. So taking these characters, he made crosses. So in one of the crosses, he made double homozygous and double dominant. For example, it is capital B, capital B, capital L, capital L. That means these two characters. Then it is crossed with double homozygous, and double recessive. So it will be capital small b, small l, small l. So what will be the F1 hybrid? If these are the parents, what will be the F1 hybrid? The F1 hybrid will be capital B, capital L, small l. That means all the F1 hybrids will be blue and long. They will be blue and long. Now if we will make a test cross, so test cross is usually made with the F1 hybrid with the double recessive. So it will be capital B, capital L, small l. This will be crossed with the double homozygous, double recessive. Now what happens? It gives rise to different types of plants. So the plants are usually blue and long, then blue and round, then red and long, and red and round. These were obtained in the proportion of 7:1:1:7. Whereas in Mendel's test cross, we usually get the ratio of 1:1:1:1. That in all these four types, both parental types and the recombinants, they are obtained in equal proportion when we make a test cross. But in this type of test cross, the blue long, so we have taken blue long and the red round. These are the parents. That means here the parental characters are appearing in more proportion, while the recombinants are coming in a less proportion, 1:1. That means here the dominant characters tend to remain together and the recessive characters tend to remain together. This is known as coupling. That means the tendency of dominant characters to remain together and the tendency of recessive characters to remain together is known as coupling. Now let us see what is happening in a repulsion phase. So in case of repulsion phase, he took single dominant and double recessive. Single dominant and double homozygous. So here I took capital B, small l. That means one is dominant, but both are homozygous. In this case also, single dominant and double recessive. And this case, small b, capital L. That means one is dominant, the other is recessive. Now when the cross is made, now it will be capital B, capital L, small l. That means again all will be blue because this is dominant character and long pollens. Blue and with a long pollen grains. Now when a test cross is made, in test cross, this F1 hybrid is crossed with recessive parent. So in this case, what is obtained, you see. Now in this case, the blue long, then blue round, then the third one is round long, then the fourth one is red and round. Now they come in the proportion of 1:7:1. Now see, blue and round, that means one dominant character, the other is recessive character, this one. And here the other one, that is red and long, then again these are the parental types. That means here the parental types are again coming in more proportion than this. Here the parental types are these two, the extreme two, 1, 7, 7, and here the middle two, they are 7, 7. These are the parental types, while the recombinants are less. So what is the conclusion from this? That means in a repulsion phase, what happens? The one dominant and one recessive, they tend to remain together, and in the other chromosome, the recessive and one dominant, they remain together. That means here the dominant characters, they tend to repel each other, and the recessive characters also tend to repel each other. That means always one dominant, one recessive, they stay together. That is the conclusion from this. So in both coupling and repulsion phase, we have seen that the parental characters are appearing in more proportion. Recombinants are appearing in less proportion. That means when in the parents both the dominant are present together, they tend to remain together, whether it is dominant or recessive. But when one dominant, one recessive there, they also tend to remain together. That means the two dominants are present separate from each other, two recessives are also present from each other. This is a repulsion, while this is coupling, and this coupling and repulsion are two sides of the same phenomenon, that is linkage. So this is about coupling and repulsion phase. Now another phase is known as cis and trans arrangement. Cis and trans arrangement. So what is cis and trans arrangement? Cis is equal to coupling and trans is equal to repulsion. So in coupling phase, what happens? In coupling phase, this capital B and capital L, they are present together, and small b, small l, they are present, that means these are present in one chromosome, while the recessive are present in the homologous chromosome. This is known as cis arrangement. This is cis arrangement. While in case of trans arrangement, what happens? In a trans arrangement, one capital B and the small l, that means one dominant, one recessive, and in the other one, that means small b and capital L. That means in one chromosome, one dominant, the other is recessive, and in the homologous chromosome, here the dominant one becomes recessive and recessive becomes dominant. So this is known as cis arrangement. This is known as trans arrangement. That means cis is equivalent to that of coupling and trans is equivalent to repulsion. So this is according to Morgan, the cis and trans arrangement, they are all equivalent to coupling and repulsion. Then after this, we'll discuss another important thing, that is the chromosome mapping. So from the linkage and crossing over, usually the chromosome maps can be constructed. How? Because in a chromosome, the genes are linearly arranged and they are arranged in a definite sequence. There are definite distances between them, and depending upon their distance, the linkage is inversely proportional. That means the lesser is the distance, the more is the linkage, or you can tell less is the recombination. So how can we determine what is the distance between two genes? Usually the distance between two genes is calculated from the percentage of crossing over. And percentage of crossing over depends upon the distance between two genes. So in case of chromosome maps, chromosome map is a graphic representation of the linear arrangement, the sequential arrangement of the genes in a chromosome, just like the arrangement of a bits on a string. And we can compare it that it is just analogous to that of a road map with some important stations. That from this station, the other station is so many kilometers. In the same way, we can, in a chromosome map, we can see that the relative distance from A to B is this much, from B to C, C to D is this much, and the sequence of arrangement of these genes one after another. All these things can be known from this. So how it is usually calculated? Usually, suppose the length between two genes may be great, but between these two genes, how many crossing over will take place? It is always not equal. There may not be crossing over. There may be one crossing over in one organism, another organism, there may be 2, 3, 4, 5 like this. That is why this is studied in a population and the frequency of recombinants is taken into consideration. So the frequency of crossing over is usually taken from the total number of recombinants, divided by total number of progenies. From this we will get the frequency, and that will give rise to the percent of crossing over. Suppose 1% crossing over is equal to 1 centimorgan. And the Morgan is after the name of the scientist T.H. Morgan. If it will be 10% of crossing over, then we can tell it is 1 decimorgan. And if it is 100% of crossing over, we can tell it as 1 morgan. This is the map unit. So how it is constructed? Suppose this is a chromosome, and suppose this is one gene, suppose it A, and it is known that the percent of crossing over between A and to B is 20%. From A to B, it is 20%. That means we have to take 20 centimorgans. So from A to B, it is 20. Then the percent of crossing over between B to C is 15%. From B to C. So from B to C, we can take sig, and this is 15%. Then suppose the crossing over percentage between B and D. So B and D, suppose it is 20%. That means from B, it will be 20%. So B up to C is 15%. That means here there will be D. That means between C and D, it is 5%, it is 20%. In this way, how the genes are arranged and which gene is present after which one, the sequence and their arrangement is determined, which is known as chromosome map. So this is how the chromosome map is constructed. Then we have to discuss what is the importance of this linkage. Now, number one, from the linkage, we can know the linear arrangement. We can know the sequence of arrangement. That means which gene is present after which one. Then we can know the strength of the linkage. Which is inversely proportional to the distance between them. And another important thing, we can predict the result of a breeding. Suppose we have made a cross between two plants having two different characters. Whether these characters will combine together or not, that can be predicted. Because if the linkage is more, then there is a less chance of the two characters combining together. But if the characters are far apart, there is every possibility that they may undergo recombination and they may recombine. So that is known as prediction of the result from breeding program. So these are the different aspects of the second part of linkage and recombination. Other things we have discussed in the previous video. Now let us discuss MCQs. So MCQs, one MCQ and 10, linkage alters the expected first choice monohybrid F2 ratio, then second choice dihybrid F2 ratio, then the third choice test cross ratio, and the fourth one is all of the above. This one question. The second question is cis and trans arrangement. Cis and trans arrangement of the alleles linked alleles of linked genes refers to their arrangement of cis and trans arrangement of the alleles of linked genes refers to their arrangement on first choice sister chromatids, second choice non-sister chromatids, then the third choice both A and B, and the fourth choice non-homologous chromosomes. So these are the type of questions. I hope you have enjoyed the video and you must have learned something about the linkage and recombination. Thank you very much. God bless you. Have a good day.